All right, so here we go. Reviewing the last of our DNA segments. Translation, as everybody who's here in the chat found out, I just rolled through like 5 minutes of this without hitting record, so I'm just going to go through real fast right now. So, translation is going to be how that messenger RNA from transcription controls the production of the protein, the polypeptide. Everything is based around explaining this diagram right here and being able to say what's going on inside the ribosome. Where are or how does that transfer RNA? What is it doing? Where did these amino acids come from? What are these connection points down here between the messenger RNA and the transfer RNA? That's going to be the important part, being able to explain what's happening here. So, keep in mind, as always, we have our central dogma and we must maintain it that DNA can self replicate virtually infinitely. But if we want protein, it's going to have to go through transcription to make the RNA. The RNA will have to go through translation, what we're talking about today, in order to make the protein. And why does that thing pop up every time that my mouse gets anywhere near the bottom half of the screen? I was going to delete this slide and not include it, but it is worth including just for this concept of the one gene, one protein hypothesis. You know, it may not be accurate, but quite often this is how things tend to work, or at least how things tend to be explained. All right, so then we must introduce our workhorse for translation, and that is the transfer RNA. This is a transfer RNA, this weird looking, kind of lobed clover leaf, I don't even know, frog foot thing. But this is one transfer RNA, and basically it's all produced just like messenger RNA in a big long strand, and then it folds over, wraps around itself. You can see these little dotted lines here because these are hydrogen bonds. And the hydrogen bonds will kind of lock the two opposite sides together in order to kind of make sure that loosely it stays in this shape. It's not perfect. You're not seeing the same kind of hydrogen bonds that you saw elsewhere in our normal molecules. But there's some form of attraction going on here. And most importantly will be our anticodon down here at the bottom in this green area, because that anticodon is what will match the messenger RNA's codon, anticodon to codon. Now, usually in a process of translation that we're writing down, you skip the anticodon part. You only worry about what is the first DNA, second, what is the codon, third, what is the polypeptide, or what is the amino acid. Now, this has to be done in our three-letter segments, which we've all worked with before. No problem there. And hopefully you guys all remember how to use our amino acid chart in order to figure out the first, the second, and the third, just to make it easier. So UAU, UA be in this box, UAU will make TYR. Now, keep in mind that every single transfer RNA that's made matches only one of the, or rather, I shouldn't say only one. There are a couple cases like that, but in general, they're designed to match one of the amino acids. It doesn't mean that there's a separate transfer RNA for every single amino acid. You can see here that there aren't. The matching to this right here, this middle box that produces the pink THR, right? There's going to be four different anticodons that all match that. All right. So, now they will then bring in the amino acids to the ribosome. The ribosome will read the codons along the messenger RNA strand. It will match up the transfer RNA's anticodons directly to the letters in sequence. And then, as this whole assembly kind of clicks through the ribosome, One by one, we add a new amino acid onto the chain. It is important to note that quite often they try to throw you by referring to the ribosome as rRNA. That's ribosomal RNA, because technically speaking, a ribosome is really just two different groups of RNA that interact in order to read other RNA. So it's kind of crazy to think about, but literally the factories that make the proteins are made of RNA just like the messages. All right, and then finally we got the sites. So the E site, the P site, the A site. I remember their order because it's like the Environmental Protection Agency, EPA, but whatever works for you works best. And what we're going to do is we're really going to go over and try to talk about what's happening at each one in order. So, kind of like everything else that we've had, we have our initiation, our elongation, and then our termination. This is no different. Initiation is going to start where the first transfer RNA that matches the start codon, AUG, takes place. That's going to land in the P subunit, and that's going to start the entire process going. Now, from there, the next matching transfer RNA will be brought into the A site, matching the next three-letter codon on the messenger RNA. Once that happens, the amino acid chain is going to be moved over one from the P site to the A site, and then the entire thing will shift down. So what was in A is now in P, what was in P gets into E, and anything that goes into the E site gets ejected. So here's our transfer RNA start match. In order to get the entire process rolling, getting ejected out, it will now find a new met and go back into circulation, maybe for use, maybe to start a new chain. That polypeptide chain will grow as each three-letter codon passes through the ribosome, one after another, after another, after another. Eventually, we're going to hit a stop codon. And the stop codon instead is going to attract this little protein jobby to come in. And when this protein jobby comes in, what happens is it kind of like, due to charges and repulsion, it kind of pushes the polypeptide chain's one connection up and out, just like a hard shoulder or an elbow just to pop it loose. Now, there are some examples, or in fact, I should say most examples that we have of bad or negative mutations are based around something that interferes with this level. Now, that interference, could it be an interference directly with the ribosomes? Yes, that's possible. Directly with the transfer RNA or the messenger RNA? Yes, absolutely. But it could also be, remember, a mutation is a change in the DNA. So if the DNA gets mutated and changed, that's going to have an effect on the proteins that you make. You won't see it right away because you actually have to go through this entire process. The DNA has to go through transcription and then translation before we see what the faulty protein actually is. So here, this phenylketonuria, right? So this is a genetic disorder caused by a mutation, and basically we should have an enzyme here. And that enzyme should turn phenylalanine into tyrosine. But if they have the mutation, they don't make the right shaped enzyme. So its active site isn't the right shape to interface with the phenylalanine. And when that happens, instead of getting converted into tyrosine, which then gets turned into all different stuff, it gets turned into an acid. As that acid builds up, it causes some pretty severe issues. You can see them down here, a lot of neurological disorders. But that's all because of one protein. Now, ultimately, that means that this is a genetic problem, but quite often it's attributed to something like translation because that's the stage where we actually see the failure. And then I want to talk just briefly about the RNA-I or iRNA, interfering RNA. It acts on transcription. And basically, what it does is it almost acts like a repressor protein to your initiation complex. It's ready to go down. It's ready to go through those genes. These sections of messenger RNA can actually wrap themselves around that, around the RNA polymerase, and just sort of gum it up, just stop it from being able to move. We're starting to study these, and things like this are really important to a lot of epigenetics because we find, kind of like the video I showed you last week, when we take a look at how we have the same traits, but they're not always expressed the same, we look for mechanisms for it. And interference RNA may be one of the best mechanisms that we have to know that maybe this is how eukaryotes go about controlling which segments of our DNA get expressed. We know prokaryotes do it from operons, something that we've already studied this year and worked on, but eukaryotes, it's a bit more complex. We're starting to get there. So, once again, that whole process from DNA to RNA, from RNA to protein, and then that protein can actually go do whatever it is that it needs to do. And all set there. All right, so let me check questions. So Matt says the anticodons are the ones that carry the polypeptide. The anticodons are the ones that carry the amino acid. The polypeptide is the final chain that's been put together at the ribosome. Yes, we use the codon for which one they produce, though, because we're reading it from the messenger, from the RNA that's going into the ribosome, not from the anticodons that are part of the transfer RNA. And if you guys take a look at the worksheet for today, it is literally practice. Like, we have all done this. When you go down to the second page, this one does ask for the anticodons. As a matter of fact, I believe we did this one in class. So it's literally nothing new. I just want to make sure that you guys first, can you pair up DNA to DNA? What should the other side of the DNA double helix look like? Then, based on that information that we get there, what would our messenger RNA transcription look like? What would the transfer RNA anticodons look like? And then, therefore, what amino acid should be produced? There is a chart here at the very bottom. Now, it's not a perfect chart by any means, but it's a little bit hard to read because it's set up such that they don't have the three-letter anticodons in here. Instead, you have to say, oh, well, if it's ACU, you go to A, you go to C, they come together in this box, and then U comes over along this top line, so we know that it's threonine. Now, you guys don't need to do this whole worksheet. Um, I just want to make sure that you get the practice going through these steps so that you feel confident on it in case they ask you a question based on it. And then tomorrow, we'll start our free response questions. As we are almost there, guys. Do you have any other questions? That's our whole review for molecular genetics all at once. Well, okay, not all at once, because we did Thursday, Friday, and now today.